Dear ladies and gentlemen, this is the third part of the talk which presents FCS measurements and their analysis. Fluorescence correlation spectroscopy is used to measure diffusion processes and concentration. The principle is easy to understand. Just imagine a single fluorescent molecule passing through the measurement volume of a confocal laser scanning microscope. This volume is very tiny, it has a size below 1 femtoliter. As soon as the molecule enters the measurement volume, the molecule is excited by the laser and a strong increase of the fluorescence intensity can be detected. As soon as the molecule leaves the measurement volume, the detected fluorescence drops. This leads to sharp bursts in the graph displaying the fluorescence intensity over time. The width of the bursts is a measure for the time the molecule has spent inside the measurement volume, the diffusion time. It would be cumbersome, however, to measure the width of each of the bursts. A better way is to calculate the autocorrelation curve, which can be interpreted in the following way. The time point at which the curve drops to half of its value is the diffusion time. The amplitude of the curve is inverse to the number of molecules inside the measurement volume. Using a calibration measurement, we can determine the size of the measurement volume. Now, we can calculate the diffusion coefficient as well as the absolute molecule concentration. FCS can be applied to measure molecule concentrations and diffusion coefficients over several orders of magnitude, for example inside living biological cells. Here we see a screenshot of the SymphoTime software. At the left side, all measurements and their results are displayed in a tree view. In order to make a FCS measurement, a point measurement is started at the laser scanning microscope at a specific location inside the sample. In the SymphoTime software, we press now Record. During the measurement, the autocorrelation and cross-correlation curves are calculated and displayed. The green curve is the cross-correlation, the correlation between two detection channels. After finishing the measurement, the curves can be analyzed. By fitting the curve, parameters like diffusion time, diffusion coefficient, and sample concentration are determined. The fitting can be extended to more complicated models, including several diffusing species, triplet photophysics, as well as restricted diffusion, to name only a few. A bootstrap analysis allows to quantify the measurement uncertainties. I now want to present you some applications for FCS. In this example, Matthias Weiss and his co-workers were interested to measure concentrations of a transcription initiation factor TIF1A inside a living cell. They co-expressed with the TIF the fluorescent protein GFP and performed FCS measurements at different locations inside the cell. On the left side, we see the correlation curves, including the fits. A clear difference in the amplitudes of the curves is visible comparing the measurements inside the cytoplasm and the nucleus. It can be deduced from these measurements that the TIF is roughly 3 times more abundant inside the nucleus compared to the cytoplasm. The measurement allows further to calibrate the intensity image in order to display the concentration in nanomolar. Thus even small local concentration changes can be visualized. The combination of FCS with lifetime, fluorescence lifetime correlation spectroscopy, has several applications. First of all, an artifact of spot detectors, the so-called afterpulsing, can be removed, allowing for more accurate measurements. Quantitative concentration measurements can be obtained by removing scattered excitation light and parasitic fluorescence. A third application is the separation of pulsed and continuous wave excitation. Using this technique, the problem of bleed-through in two-color cross-correlation measurements can be solved. The last application is the separation of two fluorophores having similar spectral properties but different lifetime. Cross-correlation measurements are therefore possible with one excitation laser and one detector. Problems are avoided which are due to different excitation wavelengths in dual-color cross-correlation measurements. How does FLCS work? If we have a look onto the TC-SPC histograms of fluorescence, after pulsing and background, and finally scattering, we see clear differences. While the fluorescence shows the typical logarithmic decay after the pulsed excitation, background and after pulsing is evenly distributed throughout the 10 nanoseconds time window. Finally, elastic and inelastic scattering, like Raman scattering, is a very fast process. It is represented by a peak at the time of the laser pulse. From these different patterns, filter functions are calculated which weight the incoming stream of photons according to their arrival in the 10 nanoseconds time window. The first photon, for example, arrives here at a very early time. It gets a negative weight since it is most probable a scattering photon. The second photon arrives at a time when the fluorescence has a high count in the TC-SPC histogram. It gets therefore a high weight of 1. The third photon arrives very late, at around 10 nanoseconds. It is most certain an after pulsing or background event and gets a negative weight. All these weighted photons are now correlated, leading to a correlation curve which is corrected from background, after pulsing and scattering. In the center we see the correlation curve of a measurement of Atto-655 dye in water. At low lag times, in the order of some microseconds and below, we observe a strong increase in the correlation amplitude. This increase is due to the after-pulsing artifact of the spot detector. The classical way of removing this artifact is to split the fluorescence equally onto two detectors and to perform a cross-correlation. Since the after-pulsing of the first detector is not correlated to the second, it is removed in the cross-correlation. FLCS removes the after-pulsing by using only one detector. In addition, also the amplitude of the correlation curve is corrected by removing background events. This leads to a quantitative concentration determination. In order to find out if FLCS works quantitatively, we have measured a dilution series ranging from 1 picomolar to 10 nanomolar. On the y-axis, the number of molecules in the measurement volume derived from FCS is plotted. At high concentrations above 500 picomolar, the classical cross-correlation technique and FLCS deliver the same results. However, at concentrations below 500 picomolar, too high concentrations are measured using cross-correlation. The FLCS technique, in contrast, obtains correct values throughout the measurement range. At very low concentrations, the curve bends. We had to use some very diluted soap solution in order to prevent the aggregation of the dye molecules onto the container walls. The soap is slightly fluorescent, leading to the small offset which results in the bending of the curve. FLCS can also be used to study cross-correlation with two dye molecules which are spectrally very similar but having different fluorescence lifetimes. In this case, the group of Petraschwili investigated double-strand DNA molecules which were labeled with two red-emitting Alexa dyes. The lifetime difference of 2 nanoseconds is sufficient to separate both dyes using the FLCS technique. The graph shows both autocorrelation curves and the cross-correlation curve, all calculated from one measurement. The cross-correlation amplitude could be determined to 70% in respect to the mean amplitude of the autocorrelation curves, showing that the majority of DNA strands were double-stranded and labeled with both dye molecules. I want to thank the groups of Matthias Weiss, Jörg Enderlein and Petra Schwille for their scientific contributions, as well as the BMBF and the European Union for their financial support. Thank you very much for your attention.